Dr. Sette. Dr. Alessandro Sette is going to talk to us today about SARS CoV2 vaccination and how it induces immunologic T cell memory and able to cross-recognize variants from Alpha to Omicron. Dr. Sette is a professor at the Center for Autoimmunity and Inflammation and the Center for Infectious Disease and Vaccine Research at the La Jolla Institute. He's devoted more than 35 years of his study towards the understanding of the immune response to multiple diseases, including cancer, autoimmunity, allergy, and of course, infectious diseases. He was one of the first groups to really characterize the T cell response to SARS CoV2, and it's therefore my great pleasure to invite him to speak to us today about the duration of our immune response to this virus. So over to you, Alex.
Before I start, I just want to point out that everything I'm going to show you today is really the result of a close collaboration between Shane Crotty and his lab and many postdocs and instructors in his lab and my lab. In particular, I want to single out Alba Grifoni, an instructor here at the La Jolla Institute, and also the lab of Daniela Weisskopf, an assistant professor at the La Jolla Institute, and many other collaborators, both inside and outside the LJI. So we started working on SARS-CoV-2 early on, and the first question that we were interested in answering was in the spring of 2020, do people that are infected develop immunity to SARS-CoV-2? And of course, as you heard already alluded to from Dr. Follman, there are different types of immunity. Antibodies, without any question, are the most important component of vaccination in terms of preventing infection. They bind the virus and basically they neutralize it. But there are also other components such as Cd4 helper T cells and CD8 killer T cells. Helper T cells are critical for antibody responses. Without a helper response, and a follicular T helper response, you do not really develop a good antibody neutralizing response. And also there was data from Stan Perlman in mouse models that really there was a protection in the respiratory tract was crucially dependent on Cd4 T cells making interferon gamma. And of course, killer T cells are important because they have a capacity to recognize and kill infected cells. So as a first approximation, antibodies are important for neutralizing the virus, but once the virus is inside the cell, it's not accessible to antibodies, which are soluble proteins, while the T cells conversely have this uncanny capacity of seeing an infected cell and eliminate it. And so they're important for modulating the infection and eventually terminating the infection. So we did this first study that eventually came out in May, and was the first study that actually detailed T cell and antibody responses in mild cases of COVID. And we wanted to look at mild cases of COVID because we wanted to have an idea of what does a success story look like? What does a situation of natural infection look like when the immune response was able to control the virus without hospitalization or severe disease? And we saw strong antibody responses against spike, and also strong cellular response against spike, which was not a given. In many other systems, the target of antibody responses is not the same of cellular immunity. So that was important because supported the notion of using spike as an antigen for vaccine development, and spike really had it all. But there were also we noted that there were strong cellular immunity responses also directed against other components of the virus, implying that if a broader spectrum of response is desired, those antigens should be considered. And this was Dr. Fauci showing our paper in Congress. It was quite humbling. And we have been sharing all our reagents very aggressively with everybody that wanted to. So what we could not answer, of course, in the early times of the pandemic is how long does the immune response induced by natural infection last? And so for that, we had to wait 6 to 8 months to see how does the immune response induced by natural infection evolve and when the immune response gets into the memory phase after the initial burst and contraction. And we saw that at the 6 to 8 months time point, still the majority of individuals had detectable and appreciable responses and multiple responses. This was actually the first study where we were able to look at T cell, memory B cell antibodies in the same people at the same time. It was the largest study for this kind for any respiratory pathogen. So this was relatively good news. At the same time, to say that 5–10% of people have low level of immune memory at 6 months implied that that is not perfect. And we were strongly advocating that people that had experienced natural infection over time should also get vaccinated. In fact, data has shown that people that have natural infection and get vaccinated develop what is called hybrid immunity, which is a awesome level of immunity that exceeds what you have with vaccination alone or with natural infection alone. Now, I want to go back for a second to the role of T cells in immunity to SARS CoV2. We had another study that came out in September 2020, where we looked at the correlation between early responses and disease outcome. And what we saw is that early CD4 T cell responses were by far the best correlate of having a mild, not severe disease. Really consistent with the notion that they, while the T cells do not prevent the infection, they can and are important in modulating disease severity. This was confirmed by a study of Bertoletti in Singapore that added on the fact that these early T cell responses actually also correlated with lower viral load. It was data from Dan Baruch in Harvard that showed that actually in monkeys, CD8 T cells are important to provide control of SARS-CoV-2, particularly in a situation of limiting antibody, neutralizing antibodies. People lacking antibodies, either because they have been depleted of B cells by in hematologic condition or in cases of MS and so forth, or that are deficient in antibodies because of genetic variation, nevertheless can control and resolve SARS-CoV-2 infection. And there's also this study looking at the kinetic showed that the onset of protection from Moderna and Pfizer vaccine tended to precede the appearance of detectable neutralizing antibodies, but in presence of detectable T cells. So the concept here is that really it's important also, as Dr. Follman pointed out, is what are we talking about, protection against infection or protection against severe disease? In talking about protection against infection, neutralizing antibodies, clearly the major determinant. But when we are investigating protection against hospitalization and death, we have to consider that there is also an important role of CD4 and CD8 T cells and memory B cells. So we recently, actually now this is published in Cell, just came out last week, we made a side-by-side comparison of immune memory to 4 different COVID vaccines. And I want to emphasize that this is important in the sense that this study was done side by side. All the people were recruited and the blood donations were given in our clinical core. All assays were done at the same time by the same people with the same instruments. So it's really very comparable. What we studied are the 2 different platforms based on RNA, the Moderna and Pfizer. The J&J is a viral vector vaccine modality, and the Novavax, which is a representative of a very classic vaccinology, a protein adjuvanted vaccine. And these donors were followed throughout vaccination up to 6 months after the first dose. So what did we find? So these are the neutralizing antibodies. And you see here the Moderna and Pfizer have a very brisk rise in neutralizing antibodies, which also wanes over time, over the 6 months period. This is a log scale. So it's about a 10-fold drop from the peak. And the performance of it, the Moderna and Pfizer, it's very similar. The J&J viral vector vaccine has a different kinetic. It continues to increase over time. It never reaches a similar level of neutralizing antibodies as the mRNA, but by the 6 months, is actually the difference is not as great as if you look at the earlier time point. The Novavax, we didn't have, unfortunately, the early time points. This was participants on a clinical study, a vaccine study of Novavax, that we were able to recruit them and obtain blood donations from them. But this was only at the later time point. But at the memory phase, you see that the level of antibodies is relatively similar for the Novavax and the mRNA. And this is the comparison between natural infection and vaccination. In all cases, vaccination was as good as or better than what is seen with a natural infection. So vaccination as good or better than natural infection in terms of neutralizing antibodies. What about CD4 T cells? Here, again, a similar picture in the sense that the two mRNA modalities are the strongest, associated with strongest CD4 responses, and then the J&J, and as expected, also the Novavax modality is not too dissimilar from the mRNA. But the one thing that I want to point out, which is striking, is that the response is much more durable compared to the neutralizing antibody, where there is a relatively rapid waning. The cellular immunity is maintained over time. A similar picture is at the CD8 level. Here, the three different platforms are fairly comparable. And again, this finding of the fact that the waning is much less pronounced, or actually in the J&J, there is no waning. And as expected, the protein adjuvanted platform did not induce a much more than a sporadic CD8 response. Memory B cells, we've seen this in the context of natural infection. We see now that again, and other people have seen this, memory B cells continue to increase in number, and the memory B cell response continues to mature over time. So really very impressive result. And so this is a graphical abstract of the paper saying that essentially we measured this different platform, and either in the case of vaccination or natural infection compared to them. And while the antibody response is waning more rapidly, the cellular response, both CD4, CD8, and memory B cells and follicular T cells, is much more maintained over time. And so this actually mirrors, or it's compatible, doesn't prove, of course, but it's compatible with the data that we're seeing. This is not our study, it's as a compilation of a Kaiser Permanente study. But I show here to make the point that the protection against infection wanes over time, while in this study was clearly noted that the protection against severe disease, hospitalization, and death really was much maintained. So what about variants? I want to bring you back to the fundamental difference in how antibodies work versus how T cells work. So what T cells do, they recognize infected cells. And how do they do that is that they recognize pieces of a virus, fragments that are derived from the virus that is inside the cells and then constantly taken apart. And this fragment of a virus binds to this HLA molecule receptor, which is very polymorphic. It's actually, HLA molecules are the one molecules that you need to match when you do transplantation. And as a result of that, while the antibody, neutralizing antibody tend to have to focus on the part of the spike protein that the virus uses to gain access to the cell and infect it, the T cells can recognize pretty much any part of spike or any part of the virus. And so that, in addition to the fact that the HLA molecules are different from one individual to the next, translates in a very large repertoire of different T cell epitopes. So in this particular study, we mapped hundreds of different CD4 and CD8 epitopes. Later on, we follow up with this other study where we compile not only the data from our own lab, but the data from everybody else in the literature. And by now, there is close to 2,500 different fragments that have been described as being recognized by T cells in humans. And why is this relevant? Because this translates into the fact that it's very difficult for the virus to mutate all different epitopes that are recognized by T cells. And in fact, we've shown this to be the case. So when you look at the response of T cells, either in vaccination or natural infection against the variants, essentially a very large fraction of the T cell epitopes are 100% conserved, and also the response is maintained in the sense, if you look at the magnitude of the response of the T cells that can recognize the different variants is in the 80% or more. But this is in contrast to what you see with the antibody response, where the mutations associated with the different variants are translate in a reduction of the capacity of neutralizing antibodies to neutralize the variants. And so we've shown this to be the case. And we also did a collaboration with several different groups sharing our reagents, and this paper from South Africa, the Netherlands, Sweden, Switzerland, really point out that this observation of the fact that while the variants are associated with a decrease in neutralizing antibodies, the T cell response is largely maintained, regardless of where you look in different geographical conditions, different patient populations. And so the concept that we are deriving here is that in an unvaccinated person, of course, as we all know, there is a wide range of presentations that vary from the asymptomatic disease to actually, unfortunately, fatal outcomes and very severe disease. And what we've seen is that vaccination is remarkably effective in preventing infection and, of course, preventing disease. In the case of a recent vaccination or vaccination protection against the wild type virus. But over time, the immune response, and specifically the neutralizing antibody, wanes, or in the case of variants, again, the capacity of the antibody response to neutralize the variant is decreased. And so this translates in a lesser capacity to protect from infection. But the fact that amongst other things, the cellular immunity still is largely retained, inconsistent with the observation that yes, the infection is not prevented as efficiently, but still there is good efficacy against the more severe outcomes. And of course, as we heard, in a boosting scenario where you raise again the neutralizing antibodies, or perhaps in conditions where you update the vaccine concept to include, for example, a spike from the Omicron variant or whatever, you would potentially go back to the situation of preventing both infection and disease. Just a couple of points here. This is a paper that is currently under review, but this is how humans look like. This is in terms of antibody response, but similar data for T cells against, in terms of reactivity against the 4 common coronaviruses that common cold that infect humans. And so you see, even in a steady state where immunity is built in the population, in the case of common cold coronavirus from repeated exposure, presumably in childhood, we reach a steady state where you have a stable plateau response and very rare reinfections every few years, perhaps, or something like that, with mild symptoms. So there is a need to continue to be vigilant. The SAVE group, sponsored from NIH and bringing together many different investigators. It's a very comprehensive approach at detecting variants, and we are part of the group looking at the T cells responses potentially against new variants. So it really needs to be stressed that all that I've been talking about could be totally upset by appearance of a new variant with completely different characteristics. And then there's the other consideration apart from SARS-CoV-2 variants. What about other potential pandemics from other SARS-CoV-2, other sarbecoviruses or other family of viruses that might be capable of triggering a new pandemic? And so these are some of the family of viruses that are a potential concern, and we are thinking of a concept where, again, looking and focusing on regions of the viral genomes that are conserved across different species, we could induce a T cell response which may not be and would not be expected to prevent infection, but would have an impact on disease severity and preventing severe disease and death. And so a broader spectrum vaccine, which we would envision, this would not be an alternative to induction of a neutralizing antibody response, but rather synergistic with that. And I just want to leave you with the this cartoon, if you want, with diagram, summarizing what I've been discussing, the fact that the protection against infection is mostly, if not all, relying on antibody responses, while the protection against severe disease is also relying on cellular immunity, both T cells and memory B cells. And this mirrors the waning of protection against infection, but the preserved to a degree protection against hospitalization and death, which particularly in this Omicron surge has been very, very apparent. And so that there are different levels of immunity, and there is a threshold that is necessary to protect against infection, mostly mediated by the antibody response, but other, such as the CD4 and CD8 and the memory B cells, play an important role. And this zone where maybe not protection from infection, but brings protection from disease, which is what vaccines are supposed to do. And with that, I think I will stop